Introduction. A new era dawns. We stand at the precipice of a biological revolution. The human genome, which took over a decade and billions of dollars to sequence just 2 decades ago, can now be decoded in hours for a few $100s. What was once the domain of specialized research laboratories is rapidly becoming accessible to hospitals, clinics, and even direct-to-consumer testing companies. By 2028, DNA science is poised to fundamentally reshape how we understand, treat, and even enhance human biology. This transformation extends far beyond simple genetic testing. It encompasses gene editing, personalized medicine, novel therapeutics, and a complete reimagining of what it means to be human. The convergence of multiple technological breakthroughs, CRISPR gene editing, artificial intelligence, advanced sequencing technologies, and synthetic biology creates an unprecedented opportunity to rewrite the code of life itself. But with this immense power comes equally profound ethical, social, and legal questions that society must grapple with as we enter this new age. The foundation. Understanding our genetic blueprint, DNA, is the instruction manual for human life. Every cell in our body contains approximately 3 billion base pairs of genetic code organized into approximately 20,000 genes. For most of human history, this code was completely inaccessible to us. We could observe traits, eye color, height, susceptibility to disease, but we couldn't see the underlying instructions that determined them. The Human Genome Project, completed in 2003, changed everything. By mapping out the entire sequence of human DNA, scientists created a reference genome that allowed us to understand genetic variations and how they contribute to traits and diseases. However, that first complete genome sequence came with an astronomical price tag and required massive computational resources. Today, the cost of sequencing a complete human genome has dropped by more than 99%, while the speed has increased exponentially. Whole genome sequencing that took weeks now takes days. Next-generation sequencing technologies have become faster and more accurate. This democratization of genomic information is perhaps the most critical foundation for the transformations we'll see by 2028. But sequencing alone is not transformation. The real revolution lies in our newfound ability to interpret and act upon this information in real time. Artificial intelligence and machine learning have made it possible to identify patterns in genetic data that humans would never detect manually. AI algorithms can now predict disease susceptibility with remarkable accuracy, identify novel disease-causing mutations in families, and even design new therapeutic proteins from scratch. The CRISPR revolution, from lab to clinic. When Jennifer Doudna and Emmanuelle Charpentier developed CRISPR-Cas9 gene editing technology in 2012, few anticipated how rapidly it would move from theoretical possibility to clinical reality. CRISPR, derived from a bacterial immune system, acts like molecular scissors. It can precisely cut DNA at targeted locations and allow cells to repair the damage in specific ways. Unlike previous gene editing technologies, CRISPR is relatively simple, inexpensive, and remarkably versatile. The first CRISPR-based therapy for sickle cell disease and beta thalassemia received regulatory approval in 2023 and 2024. By 2028, we should expect the first generation of CRISPR therapies to address a range of genetic disorders. The pipeline is substantial. Multiple research groups are pursuing CRISPR treatments for blood disorders. Sickle cell disease, thalassemia, and hemophilia represent some of the earliest successes. Doctors can remove patient blood cells, edit them with CRISPR to correct the genetic defect, and return the corrected cells to the patient. Inherited blindness. Several types of genetic blindness are being targeted with CRISPR, with some trials already showing remarkable results. Patients who were losing vision have experienced vision stabilization and even improvement. Cancer immunotherapy. CAR T-cell therapy, which modifies immune cells to fight cancer, has been enhanced through CRISPR editing. By 2028, expect significantly improved cancer treatments leveraging gene-edited immune cells. Cardiac conditions. Inherited heart conditions like hypertrophic cardiomyopathy are potential targets for CRISPR therapy, offering hope to thousands of families at risk for sudden cardiac death. However, CRISPR's path to widespread adoption faces challenges. Off-target effects, where the molecular scissors cut at unintended locations, remain a concern that researchers are actively addressing. Delivery to the right tissues remains difficult, especially for complex organs. And the ethical implications of editing germline cells, eggs, sperm, or embryos, that would pass changes to future generations remain contentious. Despite these challenges, the trajectory is clear. By 2028, CRISPR will have transitioned from promise to clinical reality, with multiple approved therapies treating serious genetic diseases. Personalized medicine, the end of one-size-fits-all treatment. For much of medical history, Doctors treated diseases based on population averages. A patient with high blood pressure received the same medication as millions of others with similar readings. A cancer patient received chemotherapy regimens developed for average cancer patients. But humans are not average. We have unique genetic variations that affect how we metabolize medications, how our immune systems respond to infection, and how likely we are to develop side effects from treatments. Pharmacogenomics, the study of how genes affect medication response, is finally moving from academic research into clinical practice. Genetically sequenced and specific mutations are identified that make it susceptible to a particular targeted therapy that would be ineffective in other cancer patients. This level of personalization is not theoretical, it's already happening in specialized cancer centers. By 2028, expect it to become standard practice. Pharmaceutical companies are investing heavily in companion diagnostics, genetic tests that identify which patients will benefit from which drugs. Insurance companies are beginning to cover these tests, recognizing that a $500 genetic test that prevents a $50,000 course of ineffective therapy is excellent value. The implications extend beyond medications. Preventive medicine becomes truly preventive when we know your specific genetic risks. If your genes suggest elevated risk for early onset Alzheimer's disease, aggressive cognitive training, specific medications, and lifestyle modifications could delay or prevent symptoms. If you carry mutations increasing breast cancer risk, enhanced screening or preventive surgery becomes a personalized decision based on your specific genetics. The synthetic biology frontier. While CRISPR allows us to edit existing DNA, synthetic biology enables us to create entirely new biological systems. Synthetic biologists are designing biological circuits that don't exist in nature, systems that can sense disease markers, produce therapeutic molecules, or even repair cellular damage. One particularly promising area is living therapeutics. By 2028, we may see approved treatments based on engineered bacteria or human cells that produce drugs on demand. For example, an engineered bacterium could live in your gut and produce insulin when blood glucose rises, essentially converting your microbiome into a pharmaceutical factory. Another frontier is the production of biologics, complex proteins that were previously difficult or impossible to manufacture. Advanced fermentation techniques and engineered microorganisms can now produce antibodies, hormones, and other therapeutic proteins with unprecedented speed and cost effectiveness. This has profound implications for drug accessibility. Medicines that cost tens of thousands of dollars could potentially become affordable for a global population. Artificial intelligence, the multiplier effect. Perhaps the most underestimated driver of transformation in DNA science is artificial intelligence. While CRISPR and sequencing technologies capture headlines, AI is quietly becoming the engine that powers discovery and application. Machine learning algorithms are identifying genetic patterns invisible to human researchers. AI is helping predict disease mechanisms from genomic data, design new therapeutic molecules, and optimize drug manufacturing processes. AlphaFold, an AI system created by DeepMind, revolutionized protein structure prediction, a problem scientists had been working on for decades. This breakthrough has immediate applications in drug design and our understanding of genetic diseases. By 2028, AI-assisted drug discovery could reduce the time and cost of bringing new medications to market by 50% or more. Personalized medicine recommendations could be delivered instantly. A patient receives genetic testing results. AI analyzes them in context of thousands of published studies and clinical cases, and within minutes, a personalized treatment plan is generated. AI is also democratizing genetic interpretation. Previously, only well-resourced academic centers could analyze complex genomic data. Now, cloud-based AI systems make sophisticated analysis available to any institution with internet access, disease prevention and prediction. One of the most profound shifts by 2028 will be the move from treating disease to preventing it. Polygenic risk scores, calculations that combine thousands of genetic variants to predict disease susceptibility, are becoming increasingly accurate and actionable. Rather than simply knowing you have a family history of heart disease, genomic risk assessment could tell you that your genetic profile places you in the highest 5% of risk for early heart attack. This specificity enables dramatic lifestyle interventions, aggressive pharmaceutical preventive therapy, and enhanced monitoring that can meaningfully reduce your actual risk. Similar approaches apply to Alzheimer's disease, type 2 diabetes, certain cancers, and many other conditions. Individuals identified as high risk through genetic testing can enroll in prevention trials or receive preventive therapies years before they would develop symptoms. The flip side is equally important. Genomic testing can identify people at very low genetic risk for conditions that run in their families, potentially sparing them unnecessary medications or surveillance procedures. The microbiome and personalized nutrition. Your microbiome, the trillions of bacteria living in your gut, plays a crucial role in your health. These microorganisms influence everything from your immune system to your mental health to your metabolism. Intelligence or athletic ability. These are no longer abstract philosophical questions, but practical ones that clinical geneticists and bioethicists must address. Social responsibility initiatives. Yet significant gaps remain, particularly between wealthy nations and developing countries. Informed consent in the era of genetic testing raises complex challenges. As the cost of genetic testing has fallen and the speed of technology has accelerated, the psychological and informational burden on individuals has grown. People might be tested for genetic variants of uncertain significance or for predispositions that may never manifest as disease. The proliferation of Consumer genetic testing has democratized access to genetic information, but has also created a landscape where people may misinterpret results or receive information without adequate genetic counseling. The use of genetic information by third parties, insurers, employers, governments remains a significant concern. While many jurisdictions have enacted protections against genetic discrimination, the regulatory landscape remains inconsistent and concerns about misuse persist. By 2028, there is heightened awareness of these issues, but gaps in protection remain, particularly around genetic discrimination in life insurance and long-term care insurance. Perhaps most contentious is the question of germline editing. While the scientific community has largely reached consensus coordination and of ensuring that advances in genetic technology benefit all of humanity, not just the wealthy nations that can afford them, the frontiers, what lies ahead. As 2028 unfolds, the next frontier of human genetics begins to emerge. The possibilities are both exciting and sobering, stretching toward futures we are only beginning to imagine. Gene drives, genetic technologies that rapidly spread genetic modifications through populations of organisms, hold promise for combating vector-borne diseases like malaria by rendering mosquitoes incapable of transmitting the disease. By 2028, these reproductive freedom, potentially allowing anyone, regardless of sex or fertility status, to have genetically related children. It also raises the intersection of artificial intelligence and human genetics is creating new possibilities. Machine learning algorithms are being trained on vast genomic datasets to identify patterns that human researchers might miss. By 2028, AI-assisted identification of novel disease genes has become routine, dramatically accelerating the pace of genetic discovery. These algorithms can predict protein function from DNA sequence, model the effects of genetic variants, and identify promising drug targets with increasing accuracy. Neanderthal and Denisovan heritage. This might be the coolest part. If you're not of purely African descent, you carry Neanderthal DNA. Between 1 to 4% of your genome comes from an entirely different human species that went extinct 40,000 years ago. Your ancestors interbred with these creatures during the Paleolithic era, not just once, but probably multiple times over thousands of years. The report tells you which chromosomes carry these ancient markers and what genes you inherited from our extinct cousins. Some people carry variations in the FOXP2 gene, a gene crucial for language, that came from Neanderthals. Think about that. You literally carry genetic code from a species that disappeared 40,000 years before civilization, agriculture, or written language existed. Rare genetic variants. Your report might reveal you carry extremely rare alleles, genetic variants found in maybe 0.01% of the global population. These rare variants often indicate your ancestors were part of particularly isolated populations, cut off from other humans for centuries or millennia. Maybe your ancestors lived in a remote mountain valley in the Caucasus, or on an isolated island in Southeast Asia, breeding amongst themselves until genetic drift created unique variants found nowhere else on Earth. The science that makes it all possible. The technology behind this revelatory power is genuinely awe-inspiring. Long-read sequencing allows scientists to read genetic sequences that are thousands of base pairs long. Imagine being able to read entire sentences of a genetic text at once, rather than just individual letters. This reveals structural variations, chromosomal rearrangements, and complex genetic architectures that would be invisible with shorter reads. Ancient DNA extraction and analysis has become so refined that scientists can recover genetic information from bone fragments, teeth, and hair thousands of years old. There's something profoundly moving about this. A person dead for 5,000 years, their skeleton crumbled to dust, Yet their DNA somehow preserved in the mineral matrix of their bones, waiting in museum drawers for scientists to finally read their genetic message. By 2028, geneticists had sequenced DNA from over 100,000 ancient individuals, a comprehensive genetic archive of human prehistory. Machine learning algorithms trained on millions of genetic variants can now identify patterns invisible to the human eye. These algorithms can process your genetic data and compare it against reference populations, identifying the most likely ancestral populations and migration routes that would explain your unique genetic signature. Massive international databases containing genetic information from billions of people. Direct-to-consumer testing companies have accumulated the largest genetic databases in human history. Academic institutions share ancient DNA. Biobanks contribute to population-based genetic data. By 2028, these combined databases represent genetic information from diverse populations across every continent, creating a comprehensive genetic map of humanity. The epic stories. DNA reveals. Here's where the real magic happens. When the data transforms into narratives, when numbers become stories of human courage, endurance, and wanderlust. The Great African Exodus. About 70,000 years ago, a small population of humans, maybe just a few thousand individuals, left Africa and began the conquest of the world. Your DNA contains the echoes of this journey. You carry genetic markers from the populations that first reached the Arabian Peninsula, then spread across the coasts of Southeast Asia. Some of your ancestors probably arrived in Australia 65,000 years ago, becoming the ancestors of Aboriginal Australians. Others ventured northward into what is now the Middle East, then fanned out into Europe, Central Asia, and eventually everywhere else. The evidence for this is written in your genes. The longer humans spent isolated in different regions, the more genetic drift accumulated, random changes in gene frequencies that happen when populations are separated. By mapping this genetic drift, scientists can reconstruct the branching tree of human migration, the conquest of the Americas. This is where DNA gets genuinely controversial. Traditional theory said people arrived in the Americas roughly 13,000 years ago via a land bridge called Beringia that connected Asia to Alaska. But DNA tells a more complex story. Yes, East Asian genetic ancestry dominates Native American populations, but some Native American populations carry genetic signatures that suggest admixture with West Eurasian populations. Some researchers even found hints of Pacific ancestry in certain South American populations. This doesn't mean Europeans sailed to the Americas in 1492 and immediately had children with Native Americans, although they did. Rather, it suggests that the settlement Middle East to Europe. Well, ancient DNA from Silk Road cities reveals something wonderful. These weren't just trade routes for goods, they were genetic bridges. People from Silk Road cities shows individual genomes that are mosaics of East Asian, Central Asian, Persian, and sometimes European ancestry. Your ancestors might have been part of this incredible cross-cultural mixing. You might carry genetic traces of a Silk Road merchant who fell in love with someone from a completely different ethnic background more than 500 years ago. Agricultural revolutions left genetic fingerprints. When agriculture spread from the Fertile Crescent, around 10,000 years ago, it didn't spread as an abstract idea. It spread because populations of farmers, people genetically distinct from local hunter-gatherers, migrated into